They'll tell you the story of muscle and nerve tissue. As usually, it should be emphasized the specificity of this particular tissue. The most characteristic feature of muscle tissue is property of contractility, which is maintained by specific arrangement of thin and thick filaments, actin and myosin filaments. There are three major types of muscle: skeletal, striated muscle, cardiac muscle, and smooth or visceral muscle. Skeletal muscle is composed of myosin plus, very long, multinucleated cylindrical fibers with diameter up to 100 micrometers. Between muscle fibers are located stems called myosatellidocytes. The sarcolemma of each fiber is surrounded by external lamina and thin connective tissue endomysium containing capillaries. Groups of muscle fibers called fascicles are surrounded by perimysium. All fascicles are enclosed within a dense connective tissue named epimysium. Here you have the microanatomy of skeletal muscle, the muscle fibers, fasciculi, perimysium, epimysium, and endomysium surrounding each particular muscular fiber. Here you have the schematized version of light microscopic representation of the muscular tissue with the endomysium subdividing each particular muscular fiber, shown by the triangular shaped arrows. Epimysium is surrounding the whole muscle, and perimysium is surrounding fascicles of muscular tissue. Here you have the nuclei, peripherally located nuclei of myosin plus, and this is sarcoplasm. Sarcos is meaning the Greek term for meat, so there are a lot of terms with beginning from the sarcos. Sarcoplasm, sarcosome, sarcolemma, and so on. Structures of skeletal muscle fibers. Internally, each muscle fiber is filled with myofibrils, composed of thousands of thick myosin filaments and thin actin filaments, highly organized into contractile units called sarcomeres. Yes, sarcomer. Within sarcomer, thick and thin filaments interdigitate. Globular myosin heads project from the thick filaments toward the fibrous actin filaments, which are associated with tropomyosin and troponin proteins. Sarcomeres are separated by Z-discs that bisect the light-staining eye bands that contain mainly the thin filaments attached to alpha-actin in the Z-disc. Between the two eye bands of a sarcomere is a dark-staining A band with a thick myosin filaments. Alternating light and dark bands appear as microscopic striations along muscular fibers. Here you have the skeletal muscle fibers with cross striations. Here you have the striations of dark and light staining. Nuclei beneath the sarcolemma and the periphery of the sarcoplasm. Here you have connective tissue endomysium. And this is particular muscle fiber, the structure and functional unit of the skeletal muscle. Here you have again light microscopic view on the skeletal muscle fibers located in longitudinal arrangement and cross-sectioned with striations, dark and white lines called striations with perimysium, endomysium, perimysium, and epimysium, fragment of loose connective tissue surrounding the whole muscular tissue. Skeletal muscle fibers and myofibrils, relationship with the fibers and myofibrils. Here you have the whole muscle. Here you have the fascicle consisting of the typical muscular fibers. Each muscular fiber, striated muscular fiber, is consisting of the myofibrils with dark and light staining bands, which are corresponding to different components of the sarcomere. And as satellite cell is fully differentiated stem cell, which can be used, which can be involved in the process of physiological regeneration of the damaged muscular tissue. Subunits of the muscular fiber are called myofibrils. Myofibrils are composed of the myofilaments. Thick filaments consist myosin. Thin filaments consist actin, tropin, troponin, and tropomyosin. Structure and functional unit of myofibril is called sarcomere. Sarcomere, the repeating sequence. Sarcos is mid, and meros is repeating sequence. Light isotropic band, I band, dark anisotropic band, A band. Z line bisect the I band, I zone, light zone bisecting the A band, and the M line located in the middle of the H zone. Repetition of these sarcomere units cause the cross relations of the muscle fibers. Here you have another view on the myofibril, consisting of myosin filaments, thin actin filaments, and this is Z line cross-linking the actin filaments, and this is M line cross-linking myosin filaments. Here is the microfilaments in cross-stained electron micrographic representation. And these are the relationship between actin and myosin filaments in different levels of this myofibril being crossed. Structure of thin actin filament is represented on this cartoon. Fibrillary actin, consisting of globular actin. G-actin is polymerizing for the formation of the fibrillary actin. F-actin, the active side of the G-actin globule is covered in the inactive state condition with the tropomyosin and troponin complexes. And here you have the relationship of thick myosin filaments and thin actin filaments, which is bisected in the M line cross binding with the specific components, keeping myosin, myofibrils, myofilaments together in the regular manner. And here you have actin, myosin with active sites covered with troponin and tropomyosin. Sarcomere is a structural and functional unit of myofibril. It's briefly, sarcomere is a segment of myofibril in between two Z lines. Thick filaments is forming central portion of the sarcomere is H zone. Thin filaments attached to the Z line extend into the A band to the edge of the H zone. I band portions of two sarcomeres on either side of the Z line. Thick and thin filaments overlap in the lateral portions of the A band. Each thick filament is surrounded by 6 thin filaments. This regular arrangement. Here you have another view on the sarcomere in between myosin and thin actin filaments bisected by the Z disc, Z line. This is M band, H band, M line, M band, A band, I band. Is half of the I band is shown to you. And the nebulin and titin molecules, which are keeping the actin filaments in proper order, in fixed position with respect to the myosin filaments. It is highly organized mode of the structural components of the tissue arrangement, which is responsible for the contractile characteristics of the muscular fiber. Here you have the different planes of cross sectioning of the sarcomere. Here you have the actin thin filament. Here is the thick myosin filament structure with the light chain and heavy meromyosin, and light meromyosin components, which are forming such globular structure, which is responsible for the movement, for the sliding phenomenon of the actin filaments with respect to myosin filaments. The rotation of the heads of the myosin filaments is moving actin filaments in the direction of the M line. Actin myofilaments are G-actin polymerized chains, tropomyosin, tropomodulin, troponin, nebulin. Myosin filaments, myosin 2 molecules consist of heavy and light meromyosin subunits, titin, alpha-actinin cross-linking protein, and CAP-Z cross-linking protein. These are structural components of molecular arrangement of the sarcomeric structural components. Here, the electromicrographic representation of the sarcomere in between two Z lines. Isotropic band, I band, anisotropic band consisting of both myosin and actin filaments, H zone consisting only of myosin filaments, and M line cross-linking myosin filaments by proteins which are keeping them in strict position, in strict manner. Mitochondria is visible, and tubulus transversum is visible. Later, we'll discuss a little bit later what is the responsibility of tubulus transversum, T-tubule. Here is another sarcomere forming myofibrils. Sarcomere, several sarcomere or several hundreds of sarcomeres are forming myofibril, and contraction, smooth sliding phenomenon of each particular sarcomere is multiplied 1,000 times because there are 1,000 times sarcomeres in the myofibril, and the whole myofibril is shortening and the whole muscle is contracting. So these are two Z lines. This is sarcomere. This is anisotropic disc. This is 1/2 of isotropic disc. This is H zone, light zone, and M line, which is bisecting the myosin filaments. These are the tubulus transversum and the sarcoplasmic reticulum forming triad of skeletal muscle fiber. Sarcoplasmic reticulum and transverse tubular system. In the sarcoplasm, between parallel myofibrils are mitochondria and cisterna of smooth endoplasmic reticulum called the sarcoplasmic reticulum, specialized for calcium 2 plus sequestration and release. At each sarcomere, two terminal cisterns of sarcoplasmic reticulum contact the deep invaginations of the sarcolemma, called the transverse or T-tubule, transverse tubule or T-tubule, forming a triad that triggers calcium ions release when the sarcolemma is depolarized. Calcium ions binding to troponin cause tropomyosin to change shape and allow the myosin heads to bind to the actin subunits, forming cross bridges between thick and thin filaments. This is basic mechanism for the muscular contraction. Here we have a sarcoplasmic reticulum and triad, tubulus transversum, two cisterns of sarcoplasmic reticulum forming triad. This is the network of the small segments of sarcoplasmic reticulum, and this is another cistern of the sarcoplasmic reticulum, and this is the orifice of the tubules, invagination of the plasma membrane inside the sarcoplasm. This is sarcomere, this is sarcomere, this is collectively termed myofibril, this is the sarcolemma, this is the sarcoplasmic reticulum with the tubule, or triad of this muscle fiber, and mitochondria, which are responsible for the energetic supply of the contracting mechanism. Here is another segment of skeletal muscle fiber showing you triads, showing you sarcomeres, showing you myofibrils in the smaller magnification, mitochondria, myofilaments, cross-section, myofilaments, nucleus of the sarcoplasmic peripheral location of the peripheral of sarcoplasm, nucleus of the master fiber, the cross-section master fiber. And here you have the external lamina, which is part of the endomysium. Mechanism of contraction. The myosin has pivot with ATP hydrolysis, which pulls the thin filaments along the thick filament. With calcium ions and ATP present, the contraction cycle ensues, in which myosin heads repeatedly attach, pivot, detach, and return, causing the filament to slide past one another, shortening the sarcomere, and subsequently shortening the myofibril, and subsequently shortening muscular fiber, and subsequently shortening muscle as the organ. When the membrane depolarization ends, calcium ions is again sequestered, ending contraction and allowing the sarcomeres to lengthen again as the muscle relaxes. Synapses of motor axons with skeletal muscle are called motor end plates, neuromuscular junctions or myoneural junctions. The neurotransmitter is acetylcholine. Mechanism of contraction, actin myosin sliding phenomenon. The hydrolysis of ATP uncouples the heads of the myosin from the actin filament. Regulation of contraction, calcium, sarcoplasmic reticulum, and the tubules. The tubulus transversum plus 2 terminal cisterns of the sarcoplasmic reticulum form a triad of muscle fiber. Calcium ions must be available for the reaction between actin and myosin, Huxley's sliding filament theory. For relaxation after contraction, calcium ions must be removed from the sarcoplasm due to the sequestrin activity. The sarcoplasmic reticulum serves as the reservoir and regulator of calcium ions. Mechanism of skeletal muscle contraction. Pivoting head of the myosin molecules, pushing the actin filaments in the direction of M-line, leading to shortening of the sarcomere and leading to the shortening of the whole muscle as the organ. Here you have the sarcomere in the relaxed condition with relatively thick H zone, relatively thick isotropic disc. And here you have the comparison of myofibril in relaxed state and myofibril in contracted state. The I bands are disappearing, the H zone is disappearing, and the actin and myosin filaments are overlapping one another for the whole length. So the sarcomere is becoming half as long as compared with sarcomeres in the relaxed condition. Here the motor end plate neuromuscular junction showing you the so-called synaptic junction between the nerve terminal and the muscle fiber. The release of the neurotransmitter is leading to the transportation of electric charge, of electric contraction signal to the tubulus transversum. This is leading to release of the calcium ions from the sarcoplasmic reticulum cisterns, and then the presence of the calcium ions and ATP, presence of ATP molecules, the sliding phenomenon occurs and sarcomere is shortening, causing the shortening of the whole muscle subsequently. Types of muscular fibers. Skeletal muscle contain fibers that can be physiologically classified as three main types. Slow oxidative type 1 muscle, fast intermediate oxidative glycolytic type 2 muscle fiber, and fast glycolytic type 2B muscle fiber, which are making different composition in the each particular organism, in each particular muscle. Then the terms, I begin to emphasizing you the terminology which is used for the description of the skeletal muscle fibers, and now the terms are fixed on the cartoon. So this skeletal muscle are related terms. Sarcoplasm, this is cytoplasm of the skeletal muscle fiber. Sarcoplasmic reticulum, this is endoplasmic, smooth endoplasmic reticulum of the muscle fiber. Sarcosomes are the mitochondria. Sarcomere, structure and functional units of the myofibril. Sarcolemma, the plasma membrane of the skeletal muscle fiber. And myofibrils are structure and functional units of the muscle fibers. Myofilaments are structure and functional units of myofibrils. Myofilaments are subdivided to actin thin filaments, myosin thick filaments. Transverse or tubulus transversum is the invagination of the plasma membrane inside the sarcoplasm of the muscle fiber. And triad, the two cisterns of the sarcoplasmic reticulum plus tubulus transversum, which is forming the initial signal, delivering initial signal for the contraction or siding phenomenon of actin and myosin filaments with one another to begin and to follow. Cardiac muscle. Cardiac muscle fibers are also striated, but they consist of individual cylindrical shaped cells, each containing one or two central nuclei and linked by adherent gap junction at prominent intercalated discs. Sarcomeres of cardiac muscle are organized and functional similarly to those of skeletal muscle. Contraction of cardiac muscle is intrinsic at nodes of the impulse generating pacemaker muscle cells of sinoatrial node. Autonomic nerves regulate the rate of contraction, heart beating. Impulse from sinoatrial node moves to atrioventricular node bundle of His, and finally by Purkinje. Fibers are delivered to the contractile cardiomyocytes. Chains of cells transducing electric impulses to contractile myocardium form conducting system of the heart. Here the light micrographic representation of the cardiac muscle, intercalated disc, the sites where the two cardiac muscle, typical contractile myocardial cells are contacting one another. Cross striations are visible. The relatively faintly staining faintly staining arrangement of the light and dark bands. The nucleus in the central position of the contractile cardiomyocyte, and lipid inclusions, glycogen inclusions in the cardiac muscle cells. No myosatellidocytes are present, and that is why the cardiac muscle tissue is completely not involved in the process of the regeneration after injury. The regeneration of cardiac muscle is being arranged by the activation of the proliferation of the connective tissue and forming of the scar of connective tissue in the cardiac muscle. Here you have the cardiac muscle diagram in three-dimensional reconstruction. The intercalated disc consisting of desmosomes and gap junctions, myofibrils consisting of the sarcomeres, mitochondria supporting the contraction of the ATP energy, the nucleus of the cardiomyocyte in the central position of the cell, and the cross striations of the contractile myocardium. Cardiac muscle is non-voluntary striated muscle limited to myocardium of heart and the proximal portions of the pulmonary veins. Consists of cardiomyocytes, contractile or typical, conducting or atypical and secretory or hormone producing. Intercalated disc contacts between the adjusting contractile cardiomyocytes. Nuclei are located in the central area of the contractile cardiomyocytes. Abundant mitochondria, blood capillaries are supporting the cardiac muscle with oxygen and with nutrients for contraction, for heart beating, which is lasting for the whole lifespan of the organism. The stops heart at 10 to 15 minutes, the neurocytes will die and the condition will be irreversible. It will be clinical death. Cardiac muscle with intercalated disc on light microscopic level, centrally placed nuclei. These dark lines are intercalated disc, and these cross striations are faintly visible, but anyhow you can recognize them. Here you have the intercalated disc on the light micrographic representation of the contractile myocardium, centrally located nucleus. Here you have intercalated disc also. Here you have the connective tissue investment of the contractile cardiac muscle with blood vessels and the lymphatics, and nuclei in the central position of the cardiomyocytes are characteristic. These are main differences, morphological difference between skeletal muscle fiber and cardiac muscle fiber. Here you have the sarcomere, Z-line, mitochondria, anisotropic disc, isotropic disc, since sarcoplasmic reticulum, tubulus transversum, lipid inclusion, thick filaments, thin filaments, H-zone, M-line, glucose, the glycogen deposits. G. Here you have the schematic diagram of the cardiac muscle with intercalated disc structure, interdigitations of the one cell contacting another cardiomyocyte, the myofibrils arrangement in the sarcomeric region, the cross striations, cross-linking of the intercalated disc structure consisting of the desmosomal junctions and gap junctions, which are arranging the cardiac muscle as the functional syncytium. Intercalated disc electromicrographic representation with some mitochondria, Z-line, isotropic disc, anisotropic disc, M-line, H-zone, desmosomes, macular adherence, fascia adherence, and gap junctions, glycogen, mitochondria, and sarcoplasmic reticulum cisterns. Cardiac muscle with intercalated disc on the electron microscopic level, smaller magnification, intercalated disc, mitochondria located in between each particular myofibril. Myofibril is consisting of the sarcomeres subdivided by Z line. And sarcoplasmic reticulum with the conglomerate of the mitochondria supplying contraction by ATP energetic material. Atrial secretory cardiomyocyte. Secretory vesicles present in the cytoplasm, in the sarcoplasm, alongside with the myofibrils, recognized due to the sarcomeric structure, mitochondria, centrally placed nucleus. So this is the natrium uretic factor producing cell. And finally, smooth muscle fibers. These are not fibers, these are individual cells, small fusiform shaped cells linked by numerous gap junctions. Thin and thick filaments in smooth muscle cells do not form sarcomere, therefore no prostrations are present. Thin actin filaments attached to alpha-actinin located in dense bodies that are located throughout the sarcoplasm and near the sarcolemma. Contraction causes cells to shorten individually. Sarcoplasmatic reticulum is less well organized in smooth muscle cells and there is no transverse tubulous system. Troponin is lacking in smooth muscle. Proteins controlling the sliding phenomenon here include the myosin, light chain kinase, and calmodulin, the enzyme which is regulating the concentration of the calcium ions in the cytoplasm. Smooth muscle is the intrinsic muscle of the alimentary canal, respiratory tract, blood vessels, genitourinary tract, and other hollow tubular organs. Smooth muscle generally occurs as bundles or sheets of the elongated fusiform cell. Cytoplasm is filled with extensive array of interviewing thin actin filaments, thick myosin filaments, as well as with intermediate 10 nanometer desmin and bimidine filaments. In dense bodies contain alpha-actinin, other Z disc associated proteins into which thin and intermediate filaments are inserted. Smooth muscles have no tubule system. Instead are present caveolae, which are providing calcium ions in the sarcoplasm. And smooth muscle is specialized for slow, prolonged, spontaneous contraction, peristaltic contraction. Smooth muscle on light microscopic level, individual spindle-like leiomyocytes of smooth muscle cells, centrally placed nucleus, cross-sectioned and longitudinally sectioned smooth muscle tissue. Here you have the electromicrographic representation of this cross-sectioned smooth muscle cell with centrally placed nucleus, with mitochondria, with collagen fibers and reticular fibers, keeping the muscle cells together in the tissue arrangement, and actin and myosin filaments cross-sectioned of filling the cytoplasm. Here you have the electromicrographic representation and the mechanism of the contraction of the smooth muscle cells. In the dense bodies, the sliding phenomenon of actin and myosin filaments in the dense bodies of the smooth muscle cells are causing the turning around the axis of the plasma membrane, and the actin and myosin filaments are attached from one side to the plasma membrane and for the other side to the nuclear envelope. And sliding phenomenon is shortening the smooth muscle spindle, making it thicker and shorter. Here is another view of the mechanism of smooth muscle contraction. This is in the relaxed condition, and this is in the fixed condition, in the contracted condition. Two smooth muscle cells, and the thick and thin filaments are sliding one with response to the other and fixed to the dense bodies. Formation and regeneration of muscle tissue. Skeletal muscle develops from myotomes of dorsal mesoderm. Cardiac muscle develops from ventral mesoderm, and smooth muscle develops from mesenchyma, the embryonal connective tissue, alongside with the connective tissue, proper and blood and other specialized tissues of the connective tissue. The skeletal muscle repair and regeneration can occur because a population of reserve myosatellite cells are existing that can proliferate, fuse, and form new muscular fibers. Cardiac muscle lacks satellite cells and therefore has little capacity for regeneration. In smooth muscle, regeneration is rapid because the cells are small and relatively less differentiated, which allow renewed mitotic activity after injury. Here, the mechanism of the formation of new muscle fibers from myoblasts and activating of satellite cells is leading to the formation of new neoblasts, which fuse with the formation of skeletal muscle fiber. Satellite cells are involved in the process of physiological regeneration of the skeletal muscle. These are terms which are related to today's topic, but to particular pathological condition of different elements of the muscular tissue. Muscle hypertrophy and hyperplasia, myasthenia gravis, Duchenne muscular dystrophy, ischemia of cardiac muscle, atheroma of cardiac coronary arteries, myocardial infarct, ventricular fibrillation, leiomyomas. You can find these terms in the internet and compare with the normal conditions which are leading what changes in normal condition of the muscular tissues leading to these pathological things in the muscular tissue. The next half of today's lecture presentation is involving the nerve tissue and the specificities of the nerve tissue from the very beginning, as usually with discussion of each particular tissue of the organism. Nervous tissue develops in an early embryo when the dorsal ectoderm neural plate folds lengthwise to form the neural tube, the precursor of the central nervous system, and releases neural crest cells, precursors for much of the peripheral nervous system, all are developing from the neuroectoderm. Nerve tissue consists of neurons or neurocytes, cells capable of generation and transduction of electric impulses, generation of electric action potentials, and neuroglia cells which support and protect neurons. Neurons are responsible for the receptive, integrative, and motor functions of the nervous system. Neuroglia includes astrocytes, oligodendrocytes, ependymocytes, and microgliocytes in central nervous system, Schwann cells and satellite mantle cells in peripheral nervous system. Each of these glial cells play a specific role in the nervous tissue function. There are many kinds of neurons, but all consist of a cell body perikaryon containing the nucleus, a long cytoplasmic extension called the axon, and one or more shorter processes called dendrites. Neurons use the common cell property of excitability to produce and move an action potential nerve impulse along the axon to excite another neuron or other effector cell. It can be glandular cell or muscular fiber or muscular cell. Dendrites are neuronal processes that receive stimuli from other cells or from the environment. Such nerve communication is transmitted to another neuron or effector cell via synapses, where a neurotransmitter is released at the presynaptic membrane and binds the receptor on the postsynaptic membrane cell, initiating a new action potential there. However, There are some specificities in the modern understanding of the axon and dendrites. Previously, it was postulated that axons are delivering electric impulse from the cell body to the effector cell. This can be the muscle or glandular cell. But today's axon has another definition. Axon is a long, slender projection of the nerve cell that typically conducts electrical impulses known as action potential away from the nerve cell body to the periphery. The function of the axon is to transmit information to different neurons, muscles, and glands. But in certain sensory neurons, called pseudo-unipolar neurons, for example, such as those in touch and warmth, the axons are called afferent nerve fibers. And the electrical impulse travels along this from the periphery to the cell body and from the cell body to the spinal cord along another branch of the same axon. Dendrites or dendrons are branched protoplasmic extensions of the nerve cell that propagate the electrochemical stimulation received from other neural cell to the cell body of the neuron from which the dendrites project. Electrical stimulation is transmitted onto dendrites by upstream neurons, usually via their axons, via synapses, which are located at various points throughout the dendritic tree. Here you have the diagram of the neuron, the perikaryon surrounding the nucleus with a prominent nucleolus and euchromatinized in most of the cases, encompassing active involvement in the synthetic processes. The axon process with terminal boutons. The axon here look devoid of the initial substance. Synaptic junctions of the axons of the other neurons with this multipolar neuron. Dendritic processes. This is typical arrangement of the most of the neurons of human body. Perikaryon, or the part of the cytoplasm which is surrounding the nuclear part of the nerve cell, because the processes are having more size cytoplasm if compared to the perikaryon, because they are long, covering long distance, either axon and dendrites. The rough endoplasmic reticulum is called the Nitzel body. It is predominantly developed in the perikaryon of the neurocytes. Mitochondria are actively supporting the machinery of the neuron with an energy of ATP. Microtubules and microfilaments are subserving for the transportation of the synaptic vesicles of the synthesized material to the periphery of the axons and dendrites. Here you have synaptic junction, here you have synaptic junction. The synthesized secretory vesicles in the cytoplasm. And of course, rough endoplasmic reticulum and Golgi complex is predominating in the neurons, responsible for the supply of with specific synthesized material. Classification of neurons includes functional classification. Sensory neurons convey impulses from receptors to the central nervous system. Motor neurons convey impulses from the central nervous system or from ganglia to effector cells. Interneurons form of communicating and integrating network between the sensory and motor neurons. Interneurons form about 99% of the total number of the neurons in human body. Morphological classification is based on the number of processes formed by the neuron. Multipolar neurons have one axon and two or more dendrites. Bipolar neurons have one axon and one dendrite. Unipolar or pseudo-unipolar neurons have one processes that divide close to the cell body into two long processes, axon and dendrite. Here you have the shape and the morphological appearance of different neuronal types. Bipolar neurons, characteristic for the retina of the eye. Pseudo-unipolar neuron, characteristic for the sensory ganglion. Multipolar neuron, characteristic for the most of the sites of the neuronal tissue accumulation. The pyramidal cell, which is called the cell of the bats, BATZ, Ukrainian morphologist, Kievian morphologist, histologist, anatomist. This is so-called pyramidal cell. And Purkinje cerebellum consists of Purkinje cells with multiple dendrites ramification, and axon leaving the cell body, leaving the cortex of the cerebellum. Here are the specific neuronal types of the light microscopic magnification. This is multipolar neuron in the Nisil substance. This is the pear-shaped cells of Purkinje. Cells of Purkinje with the Nisil substance in the cytoplasm, which is elements of the rough endoplasmic reticulum. This is neuropil, the conglomerate of the nerve cell process and neuroglia cells, and multipolar neurons surrounded with the neuronal fibers, which we'll discuss a little bit later. Neurons and neuroglia. Multipolar neuron with rough endoplasmic reticulum in the cytoplasm called the Nisil substance. The neuroglia, small nuclei of the cells of the neuroglia, which is supporting cells of the nervous tissue. And the neuropil, the accumulation of the nerve fibers of different types surrounding the multipolar neurons. Glial cells required to support neurons in many ways consist of 6 major types. Oligodendrocyte rep processes surround portions of the axons in the central nervous system, forming myelin sheaths that insulate the axon and facilitate nerve impulses. Astrocytes, the most numerous cells of the central nervous system, all produce hundreds of processes to cover and provide regulated microenvironment for neuronal pericardia, synapses, and capillaries. There are designated protoplasmic astrocytes and fibrous astrocytes, depending on their morphology. Ependymal cells are epithelial-like cells lacking basement membranes, which line the fluid-filled cerebral ventricles and central canal of the spinal cord. Microglia differ from all the other glial cells originating from blood monocytes. Remember what I told you about the macrophage system of the organism. Microglia is one of the elements of the macrophage system of the human body. So it's developing not from neural tissue precursors. They mediate immune defense activity within the central nervous system. Schwann cells or neurolemmocytes enclose all axons in nerves in the peripheral nervous system, producing myelin sheaths around large diameter axons whose impulse conductivity is augmented in the nodes of Ranvier between successive Schwann cells. Satellite cells are located within peripheral nervous ganglia. Aggregated sensory autonomic neuronal cell bodies, where they enclose each pericardium and regulate its microenvironment. So in peripheral nervous system, these cells are called Schwann cells of neuroleocytes and satellite cells or mantle cells surrounding the ganglia. You have the morphology, blind micrographic representation of the diagrammatic representation of the three-dimensional view on the oligodendrocyte and electron micrographic representation of the cytoplasm of the oligodendrocyte. Here you have the astrocytes. Fibrous and protoplasmic astrocytes are recognized surrounding the blood vessels, blood capillaries, and nerve pericardia. Microgliosides having multiple branching processes with which microglial cells are monitoring the nervous tissue of the central nervous system and looking for antigens and phagocytizing these macromolecules, delivering the antigen determinants and providing them to the plasma cells and T cells of the immune system. Ependymal cells, the cells which are lining central canal of the spinal cord and the brains of the ventricles, brain ventricles. Here you have the neuron-glial relationship. The neuron is having axon multiple dendrites. Microglial cells is protective cells of the central nervous system. Ventricle is lined by the ependymal glia, and there are so-called subtype of the ependymocytes which are called tanycyte, contacting the lumen of the filled with cerebrospinal fluid with the neuronal pericardium, making interrelationship between the fluid filled cerebrospinal fluid filled cavities with the neurons, especially for the neurosecretory neurons. Astrocytes forming the food processes surrounding the blood capillaries and the blood vessels, making the blood-brain barrier, and the pia mater cerebri, which is covering the gray matter of the central nervous system of the brain and cerebellum. And here you have the cavity of the ventricle filled with the cerebrospinal fluid. Axons in the peripheral nervous system may be described as myelinated or unmyelinated. So we are talking about nerve fibers already. The myelinated axons are surrounded by a lipid-rich layer called the myelin sheath. The myelin sheath is composed of multiple layers of Schwann cell plasma membrane wrapped concentrically around the axon. Node of Ranvier devoid of myelin is junctional zone where two adjacent Schwann cells meet. Internodal segment the myelin sheath between the two sequential nodes of Ranvier. And it is important to remember that in the long terminals of the myelinated nerve fibers, the electric impulse is jumping from one node of Ranvier to another node of Ranvier, which is increasing the speed of the delivery of the electric impulse many times fold in comparison with non-myelinated nerve fibers. Here you have this myelin sheath, oligodendrocyte in the central nervous system, covering several nerve processes. Node of Ranvier, from one to another node of Ranvier, the electric impulse is jumping, increasing the speed of the transduction of electric impulse in the organism. In the peripheral nervous system, the myelin sheath is formed by the Schwann cells. Here we have also the nodes of Ranvier. And myelin sheath is represented here and here. These are the envelopes, enveloping plasma membrane, enveloping many times fold the axon of the neuronal process. Here you have the myelin sheath formation, the translocating the duplication of plasma membrane of the oligodendrocyte, and it's increasing several times, forming several times of this bending surrounding the envelopes, enveloping the axon process. And this myelin sheath is depicted here on the light microscopic level. The myelin sheath of the myelinated nerve fibers. Here you have the node of Ranvier. Here you have the node of Ranvier. Here you have the myelin segment of one single Schwann cell. Here is the mode of formation of the myelin sheath. This is formation of the mesaxon, duplication of the plasma membrane of the neurolemocyte, thinning of neurolemocyte process surrounding several times the axon. And here the complete myelin sheath is depicted on the electron microscopic level, electron dense. This is remnants of the cytoplasm of the Schwann cell. Here you have the myelin sheath, the Schwann cell cytoplasm and nucleus, and the axon surrounded with this myelin sheath. This is surrounding with only with mesaxon. These are non-myelinated nerve fibers surrounded with cytoplasm of the Schwann cell. Myelin nerve fiber on light microscopic level, on electron microscopic level. Here is mistake, number 71 slide. This is cytoplasm nucleus of the Schwann cell, and this is axolemma, the axoplasm, the cytoplasm of the axon terminal. Here you have the structure of non-myelinated nerve fiber, mesaxon. Several nerve fibers are enveloped by one single Schwann cell. This non-myelinated nerve fibers are transducing electric impulse much slower compared to the myelinated nerve fibers, which are having jumping phenomenon of the electric impulse from one node of Ranvier to another node of Ranvier. Here, myelin sheath and node of Ranvier structure and light micrographic representation. Here, node of Ranvier and myelinated segments of myelin. Here you have the schematic representation of the Schwann cell nucleus, Schwann cell duplication of plasma membrane turning around the axon projection. The vesicles of the synaptic vesicles, which are transported to the synaptic knob, and mitochondria supporting the microtubules and kinesin and myosin with these transportation activities and microfilaments. And here you have the Schmidt-Lantermal incisures, where the cytoplasm of the Schwann cell is arrested in the mid position of the myelin sheath. Here you have the electron micrographic representation of the node of Ranvier, where the myelin sheath is not present, is lacking. Here you have myelin sheath, here you have myelin sheath, here you have myelin sheath, here you have axon surrounded or enveloped by this myelin sheath. Now we are talking about synapses. What does it mean, synapses? Neurons communicate with other neurons or with the effector cells by means of synapses, specialized junctions between neurons that transmit impulses from one neuron to another or to effector cell, which can be muscle or gland cell. Synapses between neurons may be classified as axodendritic, axoaxonic, axosomatic, dendrodendritic, and according to neurotransmitter chemistry, cholinergic, glicinergic, peptidergic, adrenergic, depending on the chemistry of the neurotransmitter. This the chemical classification of the synaptic junction is arranged. A typical synapse includes presynaptic node, the end of the neuronal process from which the neurotransmitter is released from the vesicles of the synaptic cleft, the 20-30 nanometer space between the presynaptic knob and postsynaptic membrane. And postsynaptic membrane, which has receptors for neurotransmitters, binding to which the electric impulse is transduced. The significance of the synaptic junction is one directional orientation of the reflex arc. The transposition of the electric impulse is going only one direction, the opposite direction of the typical of the electric impulse transduction from postsynaptic membrane to presynaptic knob is impossible. It's arrested. It's not having the machinery for this realization of this flow back of the electric impulse. You have diagram of different type synapses. Synaptic vesicles, mitochondria, presynaptic knob, synaptic cleft, postsynaptic density. This is axosomatic synapse. This is shaft synapse. This is spine apparatus. Axodendritic, axoaxonic synapses are also present. And here you have the neuromuscular junction, axon terminal, forming the motor end plate. Motor nerve fiber, mirror sheath, axon terminal, the synaptic vesicle containing acetylcholine, Schwann cell isolating the surface of the presynaptic knob, the synaptic cleft, the region of sarcolemma with acetylcholine receptors, nucleus of muscle cell fiber, sarcomeres, myosin filaments, actin filaments, Z-line, sarcolemma, the plasma membrane of the muscle fiber. And the synaptic junction is transposition of electric impulse from the presynaptic knob to the postsynaptic membrane, leading to the delivery of electric signal through the tubulus transversum to the sarcoplasmic reticulum, leading to the release of calcium ions as the answer to this electric signal coming through the tubulus transversum. Calcium ions are unlocking the actin and troponin junction and making the sliding phenomenon to begin. The sliding actin filaments with respect to myosin filaments is leading to the contraction of muscle elements with respect to the signal coming from the presynaptic knob. Here the motor axon may form many terminal branches, each ending at the motor end plate. Here you have motor end plate, sarcoplasm, motor end plate, the nerve fiber. All fibers innervated by branches of the axon comprise a motor unit. So this is the motor unit for several muscular fibers. Here you have the motor end plate on the skeletal, the scanning electron micrographic representation. Presynaptic knob, plasma membrane of the muscular fiber, cross striations are visible, and this forming the units of motor end plates. Here is light micrographic representation of motor end plates and electron micrographic representation of the same, showing you the presynaptic knob, synaptic vesicles, synaptic space, postsynaptic membrane with receptors for neurotransmitters, and one direction from the presynaptic to postsynaptic membrane of information of the signal is realized in synapses. Increased magnification of the electron micrographic representation of the motor end plate, the presynaptic knob, the synaptic cleft, the postsynaptic membrane, invaginations of this plasma membrane of the sarcolemma, and vesicles of this accumulating neurotransmitter, which is needed for the transduction of electric impulse through the synaptic junction. This is very small representation, but I like this segment to show you that how many synapses are possible to be arranged in one single big cell of the neuronal cell of the cortex of the cerebral cortex. And these synaptic junctions are modified through the whole lifespan, and learning, the sensory possibilities, the intellectual compounds, depending on the number of synaptic junctions which are created during the lifespan in the cortex of the brain. So this is multiple, huge amount of the synaptic junctions which are modulating the response of the organism to the signals coming from the external environment or from the inner organs, internal organs or inner substances of the organism. Development of the nerve tissue. Nerve tissue develops from neuroectoderm in the process called neurulation, beginning from day 19th of the prenatal ontogenesis. Neural tube is formed till the end of the 4th week from neural folds approaching each other and gives birth to organs of central nervous system. Neural crests located on the both sides of neural tube beneath the developing epidermis give birth to peripheral nervous system, cranial and sensory ganglia and nerves, as well as the adrenal medulla, odontoblast, melanocytes, etc. These are cells wandering of the neural crest. So you have neural to them, making the neural grow, neural crest, the notochord, neural tube formation. When the neural crests are closing, here is the segment of neural tube with neural crest surrounding neural tube from both sides. Remember what we are talking about. Somites, the notochord, the surface ectoderm, developing into epidermis. This is a general view of the developing embryo. This is cross-sectioned embryo. On the day of 1821, day of neurulation of the embryo development. Here you have the more distinct formation of the neural tube, folding of the neural plate, neural groove formation, neural folds formation, neural crest region, closing of the neural tube with respect to the notochord, with respect to developing epidermis, forming the neural crest, conglomerates and neural tube components with central canal. These are the terminology related to nerve tissue pathologies, which I advise you to look in the internet for these small designations of these pathological conditions and compare with normal state of the nervous tissue development and nervous tissue morphology. This is all I pretend to present to you today. Here are the recommended reading, and that's all for today's presentation. Goodbye, have a nice day.